See, lab diagnosis of Streptococcus, here there are two things. If it is suppurative infection, how do you diagnose? And if it is a non-suppurative infection like rheumatic fever, how will you diagnose? See, the thing with the acute suppurative infection is that you will get the pus, correct? Or a throat swab. But non-suppurative means like rheumatic fever, you will not get any kind of pus or something like that. So you have to do something different, and that is the azo titer, okay? So for non-suppurative complications, we will do what? Azo titer. We will come to the details of this later. First of all, what are you going to read? You are going to read the suppurative, that is the acute suppurative complication, how to do lab diagnosis, okay? Specimen collection, you will collect the pus, right? Pus or blood or throat swab. So you will collect the throat swab, the pus, suppurative is what? Pus or the blood, okay? You can collect from the site of lesion or leon. Collection and transport, you will use Pike's transport media. Remember the Streptococcus transport media, it is Pike's transport media. See, one thing is, you will know everything, but you can't relate them to which bacteria. That's why it becomes very hard, Strep Pike. Something you should connect it and leave it there. Strep and Pike, okay? Strep Pike is the transport media for Streptococcus, okay? Now, once you get the specimen into the lab, you're going to do a gram staining. Gram staining, you will see gram-positive foci in chains. Definitely you should not forget this word, and you should not forget to draw the diagram, correct? So you will have to draw a diagram like this, which is purple because it is gram-positive. In chains they are, and they are non-motile. As you can see, they are non-motile, okay? Now, what else? Where are we? Smear examination is important for specimens like pus. Okay, for pus there is a smear examination that could be important. Now we are moving on to culture. In case you are sleeping, please wake up. We have finished the specimen collection. We did the transport on the Pike's media. We brought it to the lab. In the lab we have done gram staining. We have seen this kind of a chain and all that. Now we have to do culture. Now remember that in classification you have already seen that it comes under beta hemolysis, correct? What does it come under? Beta hemolysis, Streptococcus pyogenes. So beta hemolysis you should see. Where is beta hemolysis seen? On sheep blood agar, it is preferred. So you can draw this type of a diagram showing beta hemolysis of Streptococcus pyogenes. This is beta hemolysis of what? Sheep blood agar, okay? Now let us move on to the selective media. So the selective media for Streptococcus pyogenes are crystal violet blood agar and PNF. PNF is polymyxin B, neomycin fusidic medium. So just we have put two diagrams here just to help you. We don't exactly have the photos of these media. Crystal violet blood agar is for Streptococcus and PNF is polymyxin neomycin fusidic acid, PNF. This is having horse blood agar. See, these two are important. Whenever you think about Streptococcus, please recollect crystal violet and PNF. Crystal violet blood agar and PNF. PNF is polymyxin neomycin fusidic acid, okay? Will you be able to remember? The problem is, I know I am saying it so many times, but you tend to know so many things, but you don't know exactly where you studied it. So say it, Streptococcus, crystal violet agar and PNF. PNF is what? Polymyxin neomycin fusidic acid. Very good. Okay, let's move on then. Where are we? Let us look at the colony morphology here. Now, colonies of Streptococci are small colonies, low convex, beta hemolysis must be present around them. and virulent strains are matte, avirulent strains are glossy. Usually something that you can remember is, if something is glossy, shiny, those things are avirulent. Why? Because they slide through. They cannot stick. Okay. So, let's move on. Just to make you wake up, I want to check where we are and how much you have understood. Tell me, what are we studying, by the way? Lab diagnosis of what? Streptococcus. In that we saw first of all specimen. See, basically if it is acute, suppurative or non-suppurative infections, then the specimen and lab diagnosis will vary. We have started with acute suppurative. In that, specimen will be throat swab or pus or blood from the site of lesion possibly. Then you will transport it. You will transport Streptococcus via Pike's transport media. Then you will take it to the lab, and when the lab you will do direct microscopy. After gram staining, direct microscopy you will do after gram staining, and then you will see what the diagram you have to draw. Then you will do culture. Culture you will do all the normal cultures, and you can do all the blood agar culture. There you will see hemolysis, small colonies, low convex. Then you will have selective media. Selective media you will have crystal violet blood agar. I think Streptococcus loves blood agar. And then you have one more PNF media, that is PNF is not Pike's, it is polymyxin, neomycin, fusidic acid, right? Let's confirm. Polymyxin, neomycin, fusidic acid, that's correct. Then we are coming to colony characters. Colony characters we saw already. Wide zone of hemolysis, small colonies, low convex. Okay. Let's continue where we left off. In hanging drop method, you should check the motility. It will be non-motile. Let us look at the biochemical reactions now. Biochemical reactions, you know it is catalase negative. You know it is catalase negative, right? Because Staphylococcus is catalase positive. And remember this blue line we have highlighted here. PYR test and failure to ferment ribose are useful in identification of Streptococcus pyogenes from other streptococci. What is this PYR test? Pyrrolidonyl naphthalamide it is. Okay, pyrrolidonyl naphthalamide is this PYR test. PYR test is going to help in identification of Streptococcus pyogenes. STR, they say it, STR. Streptococcus pyogenes is going to use this PYR test, and if PYR test is positive, then you can know that it is Streptococcus pyogenes. Now, let us move on to other biochemical reactions. Lot of them are there. I am leaving all these for you only. Now, let us see how you will identify various group of streptococci. This will come kind of under the biochemical reactions. Look at the statement here. Group A streptococci can be rapidly identified by fluorescent antibody technique. Just mark that and keep, okay. Group A streptococci can be rapidly identified by fluorescent antibody technique. So, for group A, you will identify by fluorescent antibody technique. PYR will differentiate Streptococcus pyogenes. PYR is pyro. PYR, you see, PYR, it starts with PYR only. PYRO, pyrrolidonyl aminopeptidase or pyrrolidonyl naphthalamide. Now, how will you identify group B streptococci? Group B streptococci may be identified by CAMP test. CAMP means Christie-Atkin-Munch-Peterson test. Wow. So that's for group B. What is it? Group B you will identify by CAMP test. Okay. Group D by performing heat resistance test. All the details, if you want, you can read. Fine. So I think that completes our acute suppurative. So let us revise now what and all we have read so far. Okay, great. So colony we saw. Then you will say motility, non-motile, right? Then moving on, biochemical tests. Biochemical tests, you saw that it is catalase negative. Then what else did you see, guys? PYR, that is pyrrolidonyl naphthalamide, is going to be positive for strep pyogenes, correct? This test is going to be positive for strep pyogenes. Then moving to group A. How will you identify group A? Group A you will identify by fluorescent antibody technique. Group B you will identify by, come on, wake up guys, don't sleep off. Group B you will identify by what? CAMP test, that is Christie-Atkins-Munch-Peterson test. Very good. Then group D, I have no idea, they did not mention about group C here. Group D will be heat resistance test. Okay. So we have finished the lab diagnosis of Streptococcus, at least the acute suppurative we have finished. Yay. Acute suppurative, how did we do? We saw some specific terms, Pike's transport media, crystal violet blood agar, PNF, all these are some specific words that you see only in Streptococcus. PYR again is a specific word that you see in all these. See, shall we close this? Very good. See, for Streptococcus some key words are there. Pike's transport media, crystal violet blood agar, PNF media, PYR test, CAMP test. Wow. Remember all these, okay? Tata, bye-bye. Babies, sorry to trouble you, okay? Just one more point I'll add off. See, under this biochemical classification or anything, they have added a little more, okay? You remember how the Lansfield grouping was done? Yes, it was based on carbohydrate C, okay? That is also a biochemical test you can put. And Griffith typing was based on M protein. You remember the surface protein, MTR. On the M protein, Griffith typing is done. So, please write these points also, okay? And for antigen detection and all these are standard things you should write. Antigen detection, you will do ELISA, agglutination test, molecular methods like DNA probe, nucleic acid amplification, okay? Write all this. And then don't forget to write antibiotic susceptibility testing also, okay? Antibiotic susceptibility testing. You should always send your lab diagnosis with an antibiotic susceptibility testing based with using that Kribbe-Bauer disc diffusion method, right? Hold on. That's right. It is Kribbe-Bauer disc diffusion method. See, these are standard things you will write for all lab diagnosis. Just make it a little more nice, you know, Kribbe-Bauer disc diffusion method, antibiotic susceptibility testing, right? For everything, you will get at least some marks, okay? And don't forget of all these specific blue points that I have told you. They're only there for what? Streptococcus, okay? We'll sign off now, okay? Tata, bye-bye.